The last technique we will talk about in this module is high throughput sequencing. And just like we did in a previous video, to clarify, high throughput sequencing, which is often abbreviated HTS, next generation sequencing, which is often abbreviated NGS, those are the same thing. So when you see the terms high throughput sequencing, next generation sequencing, or even massively parallel sequencing, those are all different names for the same thing. One thing that is very true is that high throughput sequencing has become much more common and important in molecular ecology studies over the years. So this graph is showing the number of results, number of publications, so that's on the y-axis, when you search specifically for the terms next generation sequencing or high throughput sequencing, and then restrict those publications that use those techniques specifically to fields related to molecular ecology. And then on the x-axis is the year. So what we can see is across time, the number of publications that use these techniques in the field of molecular ecology has drastically increased. I started my PhD roughly around here, and I had an advisor. I was lucky enough to have an advisor who recognized that this was the direction that the field was going, and he said, hey, you should do high throughput sequencing for your dissertation, and I did, and that helped me get my job here at Sac State, so I'm thankful for that. So what is high throughput sequencing? Basically, what it does is it sequences millions and millions of DNA or cDNA molecules at once. So this image here, this is actually what high throughput sequencing data looks like. What you end up getting back are gigabytes and gigabytes of files that are just these strings of nucleotide sequences, A's, T's, C's, and G's. So you're sequencing millions of molecules at once. That means you're getting tons and tons of data. So high throughput sequencing generates more sequence data than was previously possible. And because of that, you need to use techniques that are called bioinformatics, which is basically computer data analysis that has to be done with that sequencing data to be able to make sense of it all. We'll talk a little bit more about this later in the semester. There's also a course that Dr. Visker teaches here, specifically focused on bioinformatics if you're interested. But the idea is that because you're generating so much data, you have to use specific computer programs, such as packages within RStudio, to analyze all that high throughput sequencing data. So that's one of the reasons why our labs this semester are focusing on teaching you how to use RStudio and analyze data in RStudio. That's really because that's the direction this field is going. So if you're able to put on your resume that you know how to use RStudio, it will make you more competitive for jobs. So we've talked previously in this module about Sanger sequencing. What I want you to think about now is, why might this NGS or high throughput sequencing, remember, same thing, why might high throughput sequencing be better or more advantageous over Sanger sequencing? So if you remember when we talked about PCR before, what we were amplifying was one specific region of the one gene based on these primers that we designed. So at the top here is our DNA template. This is just one particular region of DNA. There's a bunch of DNA that stretches here on both sides of that template that this particular PCR is not going to be able to look at, because we're only focusing on this one particular region where the primers are binding to. In contrast, on the right here with high throughput sequencing, this blue line here is still considered our genome or our template DNA, but instead of having to focus on one particular region, because we're sequencing millions of DNA molecules at once, we are getting all of this data back. So whereas with PCR we were focusing on one particular region, in high throughput sequencing, that's not the case. we're sequencing all of these different regions at once, and that's why we get so much data back. So, beginning of the video, I said high throughput sequencing can sequence either DNA or cDNA. So one of the things high throughput sequencing can do is examine DNA sequences. We'll talk more about this later in the semester, but one particular type of high throughput sequencing is called rad sequencing. You don't need to know the details for now, but this is just an example to show you that high throughput sequencing, one of the things it can do is identify these nucleotide substitutions, which are also called SNPs, or single nucleotide polymorphisms. Again, we'll talk more about that later, but rad sequencing can identify these for us. So if you remember the example we used in an earlier video with the snails from northern and southern California. So here we have northern populations and southern populations. So what high throughput sequencing can do is, first of all, sequence multiple individuals. So that's what's shown here. Each of these lines is a different individual. And we can do what we talked about before with Sanger sequencing, where we're identifying these nucleotide differences. So in this example, in the highlighted region here, all of the northern populations have a T nucleotide, whereas all except one of the southern population individuals have a different nucleotide, a C. And so what high throughput sequencing can do is do this across a huge portion of the genome. So this is just one example from locus 1. Locus is another term for gene. So gene 1, this is one example from gene 1, but we're also looking at locus 2 and 3 and 4, etc., and literally thousands of different regions within the genome. So high throughput sequencing is a way to look at DNA sequences across the entire genome, not just one particular region. High throughput sequencing is also so popular and so important for molecular ecology because it also examines gene expression. So it's similar in concept to qPCR we talked about before, is another technique that looks at gene expression. But the difference is that qPCR looks at gene expression only for particular genes, again, based on where those specific primers are designed to bind to your template. Whereas high throughput sequencing looks at gene expression for everything. So however time point you take your sample from your organism, high throughput sequencing is going to look at all of the genes that were expressed at that given point in time in that organism. So if we go back to this example we used in the previous video, we were thinking about octocorals and how they respond to heat stress. When we were using that technique of qPCR, we were restricted to that one particular gene of HSP70. So qPCR could tell us if we expose these corals to high temperatures, how that one particular HSP70 gene would respond. But what high throughput sequencing can do is tell us what the gene expression patterns are for that organism across the entire transcriptome. The transcriptome means all expressed genes. So what we can do with this high throughput sequencing approach is we have two different experimental groups, control and heat stress. And high throughput sequencing can tell us under these conditions of heat stress, overall what every single gene is doing in that organism. So not only what HSP70 is doing, but what all the other expressed genes are doing as well. So it gives us a much more complete picture of how that organism, in this case that octocoral, is responding to heat stress. So we're looking at all expressed genes. Another example is that high throughput sequencing, or NGS, can provide information about ecological transcriptomics. So ecological transcriptomics means instead of doing a controlled experiment like we talked about in that last example, what you can do is take transcriptome samples from organisms in the field and then combine that gene expression data with things like meteorological data or other data that you can then correlate environmentally with what's happening at the gene expression level. Again, because these are not controlled experiments, you can't say for sure that these different environmental conditions like temperature or rain are causing these gene expression differences, but you can at least say that they're correlated. And so one example of this, they used high-throughput sequencing to study something called this general flowering phenomenon in Southeast Asia. So what this is at irregular intervals every so many years, hundreds of species of plants flower at the exact same time, and they didn't know what was causing this. So what they did is they took samples from one tree, both pre and post flowering. And so then with this high throughput sequencing, they were able to see what genes were changing expression in this period here. So what was happening at the genetic level, at the gene expression level, as this flowering was happening. And when they correlated that with the meteorological data, they found, first of all, that many of these plants seemed to be responding to drought conditions. And when they looked at the genes that were changing expression in these pre versus post flowering conditions, genes that were drought responsive genes were the ones that were changing expression in these individuals. So that supports the hypothesis that drought is involved in triggering this general flowering phenomena.